Okay, thank you very much. Yes, I figured, after years of COVID now, the king of respiratory viruses is back with a vengeance, and he's not happy, and he's kicking COVID's butt at the moment. So I thought this can be fairly simple. I may not tell you anything you don't already know, but I just can't let this flu season go by without talking a little bit about flu and some of the issues with it. And how we should go about diagnosing it. So, of course, some of the things you have to talk about with flu, one reason it's a problem is this whole issue of changing genome. The genome is RNA. RNA is different from DNA. DNA polymerases have a proofreading ability where when they make a mistake during replication, they can kind of back up and fix it. RNA polymerases don't have that ability. So organisms, creatures, viruses that are RNA based always are going to mutate faster than DNA viruses are. So you get this thing that we call antigenic drift. And this is not going to be unique to flu. This will be true of all the RNA viruses, so including SARS-CoV-2. So that's why you've seen all these variants of SARS-CoV-2 come along. We all know about these variants and over again. You have these same kind of variants when you talk about other RNA viruses, including influenza. We don't necessarily call them variants. In flu, they're called clades and subclades and that kind of thing. But there are all these variations, which is why you may look at it and say, I know H3N2 and I know H1N1, the hemagglutinin and neuraminidase proteins that are on the surface of the flu that we kind of group them by. But the H3N2 virus that we have circulating right now may not be the same exactly as H3N2 that was around last year or will be here next year, or the same strain that's in the vaccine. And you see this a lot also with, it's interesting with more chronic, more long-lasting RNA viral diseases like hepatitis C, where you'll actually have these waves of variants that we call quasi-species that will take place in a single patient. So just like we see all these waves of COVID that are going across the population, within a single patient, you'll see these quasi-species that will develop as it goes on and replicates over time. And then, of course, the other thing with flu is going to be antigenic shift. It doesn't have a single chromosome. It has 8 different fragments of RNA. And there's a lot of co-infections. In certain animals, co-infections with 2 different viruses are common. So when it's packaging a virus, it's very possible that you will get a mix of these RNA viruses. It will pick 3 strands of RNA from this one and 5 from that one and package them together, and then you got a brand new virus. And that is really fairly unique. You don't see that in many other viruses except for flu. Now, from a diagnostic standpoint, one thing that's problematic is that it's has a very strong seasonality. I don't know of any virus that has a stronger seasonality than flu does. It's not particularly unique. We know a lot of cold viruses, a lot of respiratory viruses are, more common in the colder climates like RSV and metapneumos. We see those, they have a season as well. And certainly the coronaviruses do. I think we're still kind of waiting to see about SARS-CoV-2 because we've been hit with wave after wave of new variants and haven't really been able to tell about a seasonality. But the kind of presumption is that once this all settles down a little bit, you'll probably see SARS-CoV seasonal in the colder months. It's not as much of strong seasonality for non-enveloped viruses. So adenoviruses in particular, they do not have this lipid envelope that surrounds influenza and SARS-CoV-2. Those envelopes make it so that they're not going to last on surfaces very long. And so they're going to be spread mainly by respiratory droplets, whereas flu is going to survive on a surface for maybe 24, 48 hours. Adeno will survive on a surface for anywhere from 7 days up to 3 months. So because they can be spread by a different pathway, they're not going to be quite as seasonable. So the problem then becomes, from a diagnostic standpoint, is that there's flu season and there's not flu season. Should you be even looking for flu outside of flu season? If you have a very low, practically non-existent transmission of flu going on in your community in the middle of July, then if you get a positive test, a positive antigen or a positive PCR for flu, it's probably not real. So you have what you do for diagnosis depends upon what time of year it is and how you interpret it. And I'll show you some data here because I love my little graphs. So here's some data that we've put together. This is from our BioFire panels, which shows you the flu A in the blue line, flu B in the red line over the past 8 years. And you can see the seasons peak very, very clearly. And in between, practically nothing. You get a few little blips every now and then. Good question about whether those are real or not. And then you get to COVID times. We have one flu B that popped up that was almost certainly a false positive. But that's it. So let me show you now. This is data from Norton Healthcare, all of our hospitals. This is all testing that was done in our hospitals using either the BioFire or Cepheid instrument for the past 6 seasons. So here's 2017, 2018. All right, there's 2018, and there's 2019. And you can see here, it's so clear all the time. December, beginning of December is when you go and get the initial surge of viruses. It kind of trails off a little bit after that. There's usually a secondary peak somewhere along the line. And then by April, it's kind of gone. And you get nothing. May, June, July, August, there's just nothing there. No flu at all. Then came the COVID year, 2021 to 2022, 2020 to 2021. There was nothing. I mean, we did thousands upon thousands of flu PCRs and never got a positive. COVID completely wiped it out. Then last year, it came on again, early December, the time that it always hits, but it was very low and prolonged season there. Probably because, we're still doing some masking and all of that. And then this season came. Boom. It's showing up 2 months early and huge, huge numbers. Let me reframe this graph. Okay. Here's what we've actually got so far this season. So we had a huge peak up here. And the good news is that it actually has come way down in the last 2 weeks. It's about half what it was. Now, when you look at graphs like this, you have to take a few, you have to stand back and look at what you're actually measuring here. What we're measuring is positive tests, not how many total tests were done. Part of the reason for this huge peak here is going to be that we're doing so many more PCRs now than we used to, because we have people who we're testing for COVID along at the same time. So we're doing a lot more. In fact, if you look at this peak right here in the 2019 to 2020 season, that was 344 positives out of 1,176 tests done. That's a 29% positivity rate. This peak, this year, 1,744 positives. So what's that? 5, almost 6 times as much. But we did 3,805. So this is actually a 29% positivity rate. This was a 46% positivity rate. And it has come down. We're doing fewer tests the past couple of weeks. It's kind of interesting, but it is down to a 30% positivity rate. So we're looking kind of like, wow, we had this massive, massive peak, but it's going down now. It's still much higher than any flu season over the past several years. And like Dr. Ramirez says, you don't know what's going to happen tomorrow. I mean, I look at this and say, see, every single one of these has a secondary peak a little bit later on. And I figure right about after Christmas, we'll probably see a second peak. And we'll see how big that is. Now, that is total number of cases. That's Norton data. If you want to look at national data, it's harder to get. It's harder for me to find total number of cases. This is the comparing years on number of influenza hospitalizations with flu, and it's a rate, the number per 100,000 people. So what this shows you is that this year is definitely earlier than any of these other years here. It's come 2 months early. What's left to figure out is where's it going to go? Is it going to slide off here and not be that terrible, or is it going to keep going way up and be much higher? And I want to show you this too, since I'm showing some national data. This is the latest data. It came out Monday morning. It's from the week ending December 3rd of all the states. Just wanted to point out, Kentucky here is purple, which is as high as they go as far as this index here. So we're as bad as it can get. If you want to avoid flu, I was going to suggest the Virgin Islands, but that's really only about 40 or 50 miles away from Puerto Rico, which is red. So you might want to avoid the Virgin Islands. So I'm kind of thinking the Northern Mariana Islands, because they're like 500 miles from anywhere out there. And in case you're interested, you can get there. A round trip ticket is only $3,000. I would recommend the one way for only $1,734. You'll save some money there. So anywhere in the United States, you're going to be hit with flu. So here's one of the charts here that shows you why this can be such a huge epidemiological issue here. This is the epicenter. These wild birds, wild aquatic birds in particular. This is where flu just gets spread around, and lots of different hemagglutinin types, lots of different neuraminidase types. And it can spread from there to domestic poultry, which spreads it around them. And it can spread to humans and to pigs. Those are the main sources where it's kind of circulating around. And lots of spread going off to other animals, including your ostriches, and I think that's a guinea pig. So it can really spread around. You can see the danger there with flu A. Flu A, you're going to have these massive epidemics every season, and the possibility of big pandemics because of the reassortment that can take place in mixed infections in these different animals and then spread from one animal to another. Influenza B, we also worry about in humans, but notice that influenza B circulates pretty much just in humans. Possible spread to a few other animals. I guess that's a seal, and that looks like a, I'm a microbiologist, so that looks like some parasite to me, but it might be a snake. Anyway, B does not have all of this going on with these different animals spreading things around, so we don't worry about B quite as much. There actually is an influenza C that spreads around in humans, and actually, if you actually go out and look for it, what you'll find is influenza C is actually fairly common. It's more common than influenza B is, but it's a very mild disease. We don't have the epidemiological issues we have with A. We don't have big epidemics or pandemics, so we really don't even worry with influenza C. But all of these people who come in with respiratory symptoms, in an outpatient setting, probably respiratory symptoms, they get tested for flu, it comes back negative. Influenza C is a possibility there. And there's also a D that probably cannot even infect humans. And just to show you these types, I think this is interesting for you. Like I said, I love looking at my graphs. This is our data from our BioFire instrument, because that's the only test that actually tells you which subtype it is. And it's just so cool to me that these viruses have a good year and have a bad year. So here's H1N1, 2009 H1N1. And in that season, that's all we had. The next season, all we had was H3. The next season, it was almost totally H1N1. And then you had a good H3 year, and then you had kind of a mix. So it's just really cool how a virus will come and say, you take your turn this year. I'm going to wait and go next year. And, who knows what that is. It might have to do with vaccine matching or whatever. But it's just pretty cool. So let's talk diagnosis here. You got a patient that comes in. They look like they have an upper respiratory virus infection. What do you do? It wasn't that long ago where basically what we said was it's a virus. Oh, you got the flu. That's one of the problems with the word flu, is that a lot of times you got the flu. And are you really sure it's the flu or not? And a lot of times it really didn't matter. You're going to go home, you're going to wait this out. If it's actually the flu, you might be totally miserable for a while. But there really wasn't much in the way of treatment. There really wasn't much in the way of anything. Just go home and take care of yourself. So we joke about it being just a virus, just a virus. But if you want to do a specific diagnosis to really show that this is true flu, what could you do? You could do it to appropriately treat the patient because now there is Tamiflu. There are some ways that you can treat influenza. There's some way that you can treat some other viruses as well. But you might have to look at it and say, is it severe enough to even warrant treatment? You might do it to manage the patient for infection control purposes. If this patient is going to be in a job or in a setting where they can spread it, do we want them to stay out of the way if they have flu? Or does it matter which specific virus it is? If I have a terrible respiratory infection that's caused by an adenovirus or it's caused by a rhinovirus, should I be staying home and not making sure that I don't spread it to other people? You want the epidemiological data, and I'm kind of guilty of this because I'm a geek. You've seen my graphs. I like this epidemiological data. I want to know this stuff. It's more important from a population perspective than it is on an individual patient perspective. So should we be testing a patient? Should we be charging them or charging their insurance or whatever for data that really benefits the population but does not really benefit them? And you could say the same thing about a whole lot of stuff that we do in microbiology. And what we see a lot these days is just to get an answer. Sometimes the patient, a lot of times with pediatrics, the parents want to know what it is. And they want the full test just to find out what it is. Even if there's nothing we can do about it, I want to know. And then the other thing you have to consider is, I use this example of an outpatient, but what does all this change to it? If they're immunosuppressed, does that change how you go about it? If they're going to be sick enough they have to be admitted to the hospital, what does that change? If this is a respiratory viral infection or a supposedly respiratory viral infection that they've had for a long time now or keeps coming back, is that something different? Or if you're going to be sending them into a setting where they can easily spread it to other people. So it gets to be kind of a complicated decision. And that's one of the things as far as diagnostic stewardship goes. One of the big questions we have is, where's the cutoff? Where's the point where we decide that we do need to have a specific diagnosis and we don't? So let's talk about how. We've got to hit viral culture because for decades, this is all we had to do. And there's a couple different ways. This is a traditional viral culture where we do cell lines that are in these tubes. We'll incubate those for up to 2 weeks and look to see if these cells get killed, basically, by the virus. So here's what a normal cell line would look like. And this is a cell line that has been infected with the virus, and the cells have died, they've rounded up. And so we would look under the scope every day for up to 2 weeks. And a good technologist could look at it and say, okay, based on which cell lines got infected, based on how long it took, based upon what that cytopathic effect looks like, I can kind of figure out what virus this is. Then we went to these are shell vial cultures. They have a little cover slip at the bottom, which has tissue culture cells on them. And you would incubate those for only about 24 to 72 hours and then use a fluorescent antibody stain. So these would be antibodies that are active against the flu A nucleoprotein. So it's not looking for actual infection effect itself, but it's looking for is expression of flu proteins on the surface of that cell. So it can be done much, much faster. But there's all kinds of disadvantages with this. Both of them have a long turnaround time. This one is shorter, but it's still long. You have to have tissue culture facilities, and not many hospitals ever really had tissue culture facilities. Only the larger ones did. And a lot of them have gotten rid of them. We do not do viral culture at all anymore. We used to up until a few years ago, but we've given up on it. We don't do it anymore. Take skill, both of them take some skill, especially the traditional. Another thing I want to point out is that don't think of this like bacterial culture. If I take an E. coli cell and I put it on a plate with the right medium, at the right temperature, the right atmosphere, that E. coli cell is going to grow and make a colony. Viruses don't do that. I could take a viable influenza virus and put it on a good tissue culture cell line with the right medium and the right temperature, and it might just decide it's not going to grow. So the sensitivity of viral culture has never been particularly fantastic. And they both are relatively expensive. So I think basically, as far as routine diagnosis, we can close the curtain on viral culture. We don't do it anymore. Very few places do them more. It's going to be done in reference laboratories. It's probably going to be done in public health laboratories. But as far as routine diagnosis in hospital laboratories, more and more hospitals are getting rid of it. Along came rapid antigen tests. We loved these things. These exploded. When these came along, they were so much easier than doing viral culture. There was just no question that you had to use these things. And there were dozens of them available on the market to choose from. Let me go through briefly how these kind of work. Here's a cartridge. Okay, it's got a membrane that runs from here down to inside this little cuvette down here. Bound to the membrane, you can't see it, but there's going to be a line here of antibodies that react against rabbit antibodies. Okay, so antibodies made by a rabbit. These might be antibodies made by a mouse that are targeted against rabbit antibodies. Here's antibodies against flu B nucleoprotein, and here's antibodies against flu A nucleoprotein. So I'm going to add the patient specimen right here, and I'm going to pretend that it has a lot of flu A nucleoprotein. That's what those are, okay? It's my little diagram. So you add that in there, and then you can't see it, but there's a pad underneath here that is saturated with rabbit antibodies made against the flu A nucleoprotein and against the flu B nucleoprotein. And they will be attached to some sort of colorimetric method, colored beads. I'm using colored beads here just as an example. I should point out there's lots of variations on this. So what I have in here is as the patient specimen migrates up into this pad, I get antibodies with the little red bead attached to the flu A nucleoprotein and antibodies attached to the little bead that are not attached to it because these are against flu B. And as that migrates, it binds here, it binds there, and you get two red lines that'll show up, a flu A line and a control line. So that's how all of these pretty much work. Like I said, we love these. They're fast, they're cheap, they're very easy to do. Anybody could do them. Viral culture, of course, could only be done in large hospital laboratories. These could be done point of care in doctors' offices. So, of course, they were used to huge extent. But the problem, we all know the problem, they're not very sensitive. This is data from the CDC, so you can blame the CDC for this. They're saying 50 to 70% sensitivity under the conditions that they're actually used. Now, you can think about 50% sensitivity. Think about like a pregnancy test. If you had a pregnancy test that was only positive in 50% of pregnant women, it wouldn't be a very good test, and you really wouldn't stand for it. But we went ahead and accepted that kind of sensitivity for flu antigen tests because we just didn't have anything else to do except viral culture. So you got to look at it and say, if you've got a patient in the middle of flu season that has symptoms and this comes back negative, you hardly believe it. You need to retest with a more sensitive method. Okay. The specificity looks okay, 90 to 95% sounds all right, but these are really not great numbers either. So if you say 90% specificity, what that means is if I take 10 people who do not have the flu, one of them is going to test positive anyway. So what that means is outside of flu season, when the chance of you actually having flu is 1 in 100,000, and you do the test and this comes back positive, it's a false positive. There's hardly any reason. In fact, the rule was don't use these outside of flu season. Don't use them because the specificity was too low. I have always had problems with outpatient clinics that use these, and they would call me up and say, it's the middle of August and we've already had 8 positive flus. How come you're not seeing this? And I'm like, oh, you shouldn't be doing that test in the middle of August because there's no flu around. Those are all false positives. So the FDA got involved with this. Back in 2017, they passed some new regulations which said we're going to reclassify these rapid antigen tests. We're going to require that manufacturers show that they can get at least 80% sensitivity compared to PCR or 90% sensitivity compared to culture. And they have to get a specificity that's at least 95%. On top of that, every year, they need to test against viruses that are currently circulating. The CDC provides a panel of viruses. These are the ones that are currently circulating. They have to be able to show that they can detect them with good sensitivity and specificity. They had until January 2018. So a lot of the rapid antigen tests that were on the market in 2017 were no longer on the market after 2018 because they couldn't go through and do this. One of the problems I should point out with the circulating viruses, a lot of these tests were developed in 30, 40 years ago. And they worked against the viruses that were circulating at the time. But the antibodies that they used were never updated for current viruses. So they may have had 90% sensitivity in 1970, but those same antibodies, by the time they got to 2018, may have been 50% sensitive. So now they have to make sure that they can go against viruses that are currently circulating. A lot of tests came off the market. The ones that are more popularly used now are like these guys right here. Still the same principle with these, but they are read by a machine. The Veritor, the Sophia, these are probably the two most common ones that are used now. So having the machine read it gives you better sensitivity and better specificity figures. And I'll just show you here, this is an example of what they have to do every year. The CDC provides a panel of 8 different viruses, and they have to go through and show that they can detect those at a reasonable concentration. To stay on. I'm going to skip that just because we have to get to molecular in the last few minutes. So we've all gone to molecular now. The antigen tests are becoming less and less often used because we have molecular tests. The reason we switched to these, they're better. Sensitivity is better. I say 90 to 99, but really you're talking about 95, 97, 99% sensitivity. The specificity is better. So you can do these through the whole year, but you still have to be very wary of one that gives you a positive test when there's no flu circulating. That's probably a false positive test still. So they're just much better tests. I mean, there's no contest there. It's easier to multiplex. The antigen tests don't multiplex very well. So now what we're doing, the reason we're doing so many flu tests is because we have them in panels. So you test flu A, flu B, and RSV at the same time, or flu A, flu B, and SARS-CoV-2 at the same time. And so most of the flu tests that we're doing right now are actually multiplexed with other respiratory viruses at the same time. That's easier to do with molecular than it is to do with an antigen. And believe it or not, money came into play as well. Yes, these things cost more, but you get better reimbursement. So the companies that made the molecular assays would come to a laboratory and say, look, you're going to pay more. I'm making up these numbers here. This is about what an antigen test would cost. This varies a lot from one facility to another, whereas a molecular test might be 2-3 times the cost. But if you're going to get reimbursed, and most of the time these are going to be outpatient, so you will get some reimbursement. The reimbursement is much better for a molecular assay. So you actually come out ahead by switching to molecular than you do with staying with antigen. Then things got much easier. The old-time PCR is almost gone. Now we have a much more automated PCRs available. So this is what drove point-of-care sites to actually start it. This is a big Cepheid instrument that might be in a large laboratory. Very easy to do. Now Cepheid has got a waived version that can be done in doctors' offices, immediate care centers. We have several of these in our a couple of these in our emergency rooms. They use the exact same cartridge. It's just, this is for lab, this is for, it's really a difference in the software. Is really, the modules are doing exactly the same thing. It's a difference in the software that lets it be clear wave. Same kind of thing. This is a big honking Roche instrument that does tons and tons and tons of these all at one time. Our outpatient facilities use the Roche Lyons, which runs a single one at a time, but obviously on a much smaller scale. And then there's the panels. A hospital laboratory may use a big torch instrument that can have anywhere up to 12 modules in one instrument. But they also have a waived version of this respiratory viral panel that can be done in a hospital, a clinic, a point of care site. for very little effort. So lastly, since we got to this thing about these panels, do you have to run the big panel? Do you need the big panel? This is the BioFire panel over here, the most popular one that's being used. And the answer is no, especially if we're talking about influenza. If you want to diagnose influenza, no, don't use the big panel. The big panel would be helpful if you need to identify a virus other than flu, COVID, or RSV, the viruses that we can detect with easier, simple, smaller panels on our molecular instruments. Is identification of some other virus going to change patient management? If a patient comes in, do you care if they have parainfluenza virus 2, or do you care if they have coronavirus HKU1? Is that going to change how you manage that patient? Those are the only situations where you should be using the big panel. Now, the big panel for flu does have some advantages. This is the way that we can subtype. Notice in here it has H1, H3, and the 2009 H1N1. So we can subtype flu A with this, which is helpful sometimes. And theoretically, it can help detect new strains of flu, because we will get some every now and then that are non-typable. It's a flu A, but I can't tell whether it's H1 or I can't tell whether it's H3, so it's non-typable. Those we send to the state laboratory, and they can go a little further with it, and they can send it on to the CDC if necessary. So if we did have a situation where somebody, came down with H7N1 or some other strange variant of flu, that's how we would detect it here. We would think it's something weird, and we would send it to Frankfurt, and Frankfurt would figure out, yes, you're right, it is something weird. The money, I'm going to say 150. It varies a lot depending upon the size, how many of these you buy, that kind of thing. But these are considerably more expensive. And I can spend 3 hours talking to you about reimbursement and still not understand it. There's a whole lot of questions going on right now about whether these are going to be reimbursed. So let's end up here. Just the progress that we've gone through for influenza. Viral culture only in hospitals, pretty much obsolete now. The rapid antigen testing started in hospitals, moved to the point of care setting pretty quickly. We moved to kind of a very manual, intense molecular, which has now become a kind of automated molecular in hospital laboratories. That is now available in point of care. The fact that we're doing PCR in doctor's office, in a pediatrician's office, in an emergency department, that's crazy. I mean, you never would have thought that just 5 years ago. And those are all kind of things that have really been pushed so much farther along with COVID. Now, the next step is going to be flu. I skipped over that slide, but we don't have any home test for flu. There is no home antigen test for flu. There will be soon. And there will be molecular PCRs being done in the home. I don't mean being collected in the home. I mean actually being done in the home. Those are going to come along. It's probably going to take about 5 years before we get to that. So we're moving toward faster, easier, and point of care / home testing for flu. Now we sit around and we wait to see what's going to happen with this flu season, because you don't know what's going to happen tomorrow. We may have another group. Anyway, that's all I've got for you. So didn't go a little bit over. Be happy to take any questions if somebody's got something.
We'll see if we have any comments or questions from the audience. I think that David, we don't have any comment in the chat, no?
No, sir, we do not.
I mean, you, Alan, again, a great summary. I have a long list of things, but how is that we're going to be able to do PCR at home? How is the?
Yeah, I know. I'm not very creative in this way, but there is a company that's working on it. It's possible. One of the things that might happen is that is not PCR per se, because PCR, when we think of PCR, you have to have a thermal cycler, and that's going to be difficult to do. But there are other amplification methods Like, for example, one of the ones that we use here on some things is transcription-mediated amplification, which is all same temperature. You do not need a thermal cycler for that. So there are other amplification methods that might be more amenable to a simple little strip where all the stuff that can be done inside, and then there's some kind of a color signal or something that will come up with it. So it can be done. You know, there's a lot of things that have happened in the last 10 years that I thought were stupid. That's crazy. That's never going to be accomplished. And it got done. So you don't want me running your venture capital firm.
Yes, yes. There is a form of amplification of the RNA in a way that is, you don't need, up and down temperatures, or it's
Right,
just
yeah.
in the key. Perfect.
Yeah.
Perfect.
Yeah, we tend, we always kind of use the word PCR,
Yes.
but we shouldn't. PCR is one form of amplification. There are lots of others.
Yes. There is in the case of the bacteria, we're always interested in the you give us 1 plus 2 plus 3 plus. There's always some form of quantification.
Yeah.
And for and or sometimes you give us you give a presentation also where you can give us the actual colony count, and then you can say 100,000. Do we have any? It doesn't make sense, or because sometimes we get from sometimes we get a quantification of a viral load or whatever we want to call it. That this is something that how this is done and how you see this, that this makes sense.
Yeah, okay, that's great. Yeah, there are the viral loads that we do. It's by a molecular method, but they're meant to be used in situations where we already know you have HIV. What we want to know is how much HIV you have. And there's specimens that are relatively uniform in collection. It's going to be plasma. So you stick a needle in somebody's arm and draw some blood. Okay. With flu, we'll use flu as an example here. The key thing is you have flu. So how much flu you have doesn't matter. It's kind of like a pregnancy test. We don't care how pregnant you are. You're pregnant. If you have flu, you have flu. Now, following it over time, we don't really do. The other issue with flu in other specimens like that would be there's more variability in specimen collection. I mean, I know good and well that when we ask for a nasopharyngeal swab, sometimes we're not really getting a very good nasopharyngeal swab. So if you see a decline in viral load, you say this person here doesn't have much. You have flu, but you don't have a whole lot. Well, is that because they do not have a heavy infection, or is that because it was not a particularly good specimen to collect? So there's going to be more variability with specimen collection with those. So is it possible to do a viral load on flu? Sure, it would be possible to go through. You're going to have to go through all the stuff of making, having, first of all, standards to compare it to. I would have to be able to purchase or create myself. Here's a suspension of flu A virus that is at 100 copies per mil, and 1,000 copies per mil, and 10,000 copies per mil, and then be able to compare my patients to that. So it would be a heck of a lot of work, a lot of supplies that would be needed for it. And I think people look at it and say for respiratory viruses, for acute respiratory viruses, it's not going to provide you with that much useful information. It would be cool.
Yeah, no, yes. I have another question. Then you mentioned that if the patient is negative, but you suspect that the patient has the disease, you always question, was the nasopharyngeal swab done appropriately? And again, we come out from the nasopharyngeal swab from the pediatric data into the adults, and now with COVID, everybody was having the nasopharyngeal swab. But then looking at the rapid test at home, then of course nobody's going to be able to do a
Nobody's going to do that.
self-pharyngeal swab. Then how do you see, and of course we're doing some research collaboration looking at this, but how do you see the sample? Because at home everything has to be simple. But then to see if I have COVID or not, I just swab my nose or get saliva. And why is that if I come to the emergency room, I do a nasopharyngeal swab? You think that is so much of a difference, or everything is, I mean, the sample type to see if you have a viral respiratory infection, is this critical, or we're still learning and
I think we're learning that. I think, it's basically inertia. We have always used nasopharyngeal swabs. And they have switched, especially with some of the point of care ones, to nasal swabs. They work as well. Sometimes they work better, like RSV in kids. Kids that have RSV have snot oozing all over the place. And it's easy to huge volumes. So specimen collection there is not that difficult. But in other situations, you're not going to have as much profuse material leaking out of somebody's nose. But they've gone to simpler methods like a nasal swab. But I think, we developed that nasal swab, we developed that viral transport medium back when it was viral culture was the way to do it. And you could even, I'm making stuff up here, so don't really consider this correctly, but you can even kind of see if the site of infection is the nasopharynx, then viable virus will be found there. What you get from the nose might not be viable anymore. So it wouldn't work as well for viral culture. It would still work for antigen. It would still work for amplification. So I think it just takes us 30, 40 years to kind of switch these things, and we need to do, we can look at different specimen types and say, does saliva work? Does a cheek swab work? Does a nasal swab work? There just needs to be the impetus to do that kind of study. Meanwhile, we're stuck in laboratories, and especially in point of care settings, because in a point of care setting, you have to follow the manufacturer's instructions to the letter. You must do exactly what they say. So until that manufacturer validates nasal swabs or saliva, you're stuck. You cannot do it.
Very good, very good. I think that it's 12:45. This is the time that we mentioned for everybody that we are going to extend. This was another a great presentation, and I want everybody to see that for example, we've been discussing flu from different angles. Recently we have a flu more like for an infection prevention. This was a flu more microbiology, and I think that this is the way for us to learn these topics, from the same topic from different angles, and then we know what to do based on our different activities. Alan, these have been great. Thank you very much. And I'm sure that this group doesn't need to be told that we need to get the flu vaccine, but just for all of us to remember, looking at these numbers, anyone that we know still is a good time to get vaccinated this year. There's a lot of flu. Alan, thank you very much. Thank you, and we'll see you this coming week.